Hello, everyone. It's great to be here. I'd first like to really thank the organizers of Pittcon, and especially, of course, the trustee beyond the Wallace Coulter lecture. It's a real true honor for me to receive this award, and it's also a great pleasure to present this presentation. Of course, ideally, I would like to do this in front of a real audience. But given the circumstances, I'm really happy that we can still present this lecture. And I'm happy that we can do it now online, and I hope you enjoy it as much as I do. So because it is online, I also thought to make the format slightly different. So I will actually present my lecture in 3 parts, 2 more scientific parts, and 1 more science meets art part. And I think we can actually record and take these 3 sessions and then have little breaks in between if you want, or you can all watch it in one go. So the 3 parts that I will talk about is sizing and counting particles by native mass spectrometry. The second part is how unique is your immune response. And the third one is a nice video that we made together with some artists about our research. If you get an award like this, you always think, okay, that's very nice, recognition for my field, recognition for mass spectrometry. And sometimes you give them a lecture that is a bit more historical, talking about your career, about your past. But actually, I really want to talk about the most recent things that we have been doing in our lab. And that's why I did choose these topics that you see over here. But that doesn't mean that I don't want to look a little bit at history. And of course, this lectureship, the Wallace Coulter lectureship, is named after Wallace Coulter, and I think you know him all. Of course, very famous, also of Beckman Coulter, but also, of course, of the Coulter counter or the Coulter principle. And I really like what I found on the internet about the Coulter counter, also the principle. So the Coulter counter is very important. It's now basically used in almost every hospital throughout the world. And it's really a device, and you see a sort of schematic of it here on the slide, that particles, and these particles could be macromolecules, but it could also be cells, they go into a little hole, and by going through this hole, they actually change the impedance of the device, and that is measured. So each particle that moves through this changes this current, and therefore it can be detected. And how much it changes this current depends on the size of the device. So it's a particle counter, but it's also a particle sizer. And of course, you see now in the middle, you see the original patent, beautiful pictures, maybe even made by Wallace Coulter. And I like this device because part of the talk where I will talk about is mass spectrometry. And I will talk also about how and if we can measure single particles by mass spectrometry. And then maybe can also size them and also count them. So maybe I want to talk a little bit about how a mass spectrometer can act as a Coulter counter. So that's sort of my maybe relationship to the Coulter award, but also relationship to a bit historic perspective. So sizing and counting particles in the gas phase, that's of course the difference between the Coulter counter and mass spectrometry. Now I see this award and also Pittcon as a great analytical conference. And mass spectrometry is of course an important technique in analytical chemistry. And mass spectrometry is something that I've been working on for the last 30 years or so. And mass spectrometry is a very big field. My field is biomolecular mass spectrometry. I'm mostly interested in analyzing proteins and peptides. And for years, we have done that in many different flavors. So a big part of biomolecular mass spectrometry is actually proteomics, is to analyze and quantify and identify proteins from cells and tissue. And we have a workflow in that has been developed over the last decades that is now very sophisticated. We have done that, but many other labs as well. And at the moment, we can really use these mass spectrometry-based proteomics techniques to look at pure proteins, purified assemblies, but also at very complex mixtures like whole cells, whole tissues, and whole organisms. And we do this mostly by getting the proteins out of these tissues or lysates, then digest them, and then bring these peptides to the mass spectrometer for identification, quantification, sequencing, etc. Now, I don't want to talk about that, but what I just want to show is that mass spectrometry and the techniques used in mass spectrometry can also go way beyond these proteomics applications. And some of the applications that I find particularly interesting is where we can use mass spectrometry not only to identify proteins, but also to look at protein structures, protein function, protein interactions. And we use the same technique, mass spectrometry, but in slightly different flavors. Because if we want to use mass spectrometry for structural and molecular biology, we think more about techniques like chemical cross-linking coupled with mass spectrometry, hydrogen-deuterium exchange coupled with mass spectrometry, limited proteolysis, or by looking at intact proteins or even intact protein assemblies. Now, of course, this was a field that matured much later than proteomics. And first of all, we thought, is it ever possible to get whole proteins and protein complexes inside the high vacuum of the mass spectrometer and keep them there under conditions that we still call sort of physiologically relevant or native? Now, this is a field where my lab has been very active in, and I just want to show you a little bit about that and also then how we see the future in that field. Now, the field where I'm talking about is also native mass spectrometry. Now, I just have one slide to explain that a little bit. Normally, when we do mass spectrometry and biomolecular mass spectrometry, we often couple it to liquid chromatography. But liquid chromatography, as beautiful as it is, often occurs in a fluid that contains an organic, like acetonitrile, and it's often also acidified by formic acid or something else. And this really takes in, or the consequence is that the proteins that you analyze by LC-MS, they really get denatured. And by becoming denatured, they also become, in this acidic pH, highly charged. And therefore, if we analyze proteins by standard LC-MS, we get of a single protein a lot of different charge states. As you can see here in the sort of brownish block, enormous amount of charge states at low M/Z. The protein is fully denatured. It's not functional anymore, of course. And what we have been working on is to see if we can actually keep the protein more in its native state. Now, we do this by not using the organic element, also by keeping the pH at about 7. And if we do that, we can actually spray these proteins directly in the mass spectrometer. And what you see in the blue box is the same as what you see in the brown box, but the same protein analyzed by either denaturing MS or native MS. Now, and if we do it by native MS, what we see is that the protein picks up much less charges because it is still folded and much less of the proton acceptors are available. And that's one of the reasons why we think the protein is still more physiologically relevant, more native. Now, what you see, this native and denaturing MS for purified proteins, it makes not such a big difference if we want to do a mass analysis. But most proteins in life, they are not simple proteins like at the top. Proteins in life, in cells, they are often modified by modifications like phosphorylation, glycosylation. And on this slide, you actually see two examples of a phosphoprotein and a glycoprotein. And just to look directly at the bottom, we see the glycoprotein. We see the charge states. There are, again, only 3 or 4 different charge states. But every peak in this spectrum is the protein with a different glycosylation. Now, if you measure that by native mass spectrometry, you can see these different glycans attached. You can measure these glycans attached, and you get a whole glyco or proteoform profile of this glycoprotein. But here, if you would turn back to denaturing MS, the standard MS approach, you see the spectrum on the left, and it becomes too congested to really get the details of your protein. So this is one of the nice examples of native mass spectrometry. But native mass spectrometry, as I said, had to mature compared to proteomics. And one of these maturation steps was that we need mass spectrometers that can really measure intact proteins more sensitively, and that can also measure them under more native conditions. Now, you see already on this slide that one of the things that we had to modify is to get the M/Z range of the mass spectrometers extended, And the other thing we had to modify is to get the transmission of these very large particles with relatively little charge, to get this transmission through the mass spectrometer much better. Now, I will not go into too many details, but we worked on this for several years, also in close collaboration with Alexander Makarov, the inventor of the Orbitrap mass spectrometer. And what we did is, together with him, we modified his Orbitrap instrument to actually allow the better transmission of high mass ions, and also to allow detection at much higher M/Z. We did that over a period of 10 years. And we also made prototypes, but then these instruments were also commercialized. And so the first Orbitrap that was really amended to work for high mass ions, we made together, but was then commercialized as the Orbitrap EMR. And EMR stands for Extended Mass Range. And then a couple of years later, we made quite a big improvements. And this instrument is now commercialized under the name Orbitrap UHMR, standing for ultra high mass range. Now, the modifications, I will not go into detail, but they were all centered around changing the voltages, the RF fields, also changing the quadrupole. And with that, we could really get the transmission of high mass ions much better. Now, just to show you some examples of what is possible, I first want to state, when we published this first paper about the EMR, we said this can really be a game changer. Now we can really open up a whole new field and look at intact proteins, intact protein complexes, and maybe not only proteins, but also very, very large macromolecules like ribosomes, like viruses. And we proposed this in 2012, and now about 10 years later, we have seen that this is really has become true. Now, just again, one simple example of one of the first native mass spectra on the Orbitrap, a very important example, because this is a mass spectrum of an antibody. Now, of course, mass spectrometry plays now a central role in the quality control procedures of all biopharmaceuticals, all modern biopharmaceuticals. And in this spectrum, what is so nice is that we see this 150 kilodalton antibody in a high-resolution mass spectrum. And we see, if we look at the charge states and we zoom in, we see not only the charge states of the antibodies, but we really see in the details the glycosylation pattern of the antibody. We see also in the minor peaks that you see on the left and the right of the main peaks, you see some small impurities, maybe from lysine clipping, but we can really measure these masses extremely accurately, and we can measure these and identify and quantify then the glycan composition on the particles. Now, this was really a game changer, and now this instrument and native mass spectrometry is used more and more to characterize antibodies. But that field of producing biopharmaceuticals is also rapidly developing, and I just want to make a big step forward. On the left here, you see the antibody in A, but you see all kind of new biopharmaceuticals appearing, used also in the clinic. Fusion proteins, maybe coupling of antibodies to make bispecifics, and even more so. And also some antibodies, they have on purpose been made into hexabodies. And this is something that we have a collaboration with one of these biopharmaceuticals, Genmab in the Netherlands. And you see that if you make these molecules more and more complex, and I guess you agree that the molecules in B, C, D, F are way more complex than A, the quality control is getting more and more difficult because you produce these compounds in cells. And of course, the more complex your molecule is you want to make, the more complex also maybe the side products are, and you need to analyze those as well. Now, they not only become more complex, they also become larger and larger. And what you see over here is a mass spectrum that we recorded quite a few years ago, of an hexabody of IgGs. So that's really already a molecule of about 1 million Dalton. And we then measured also the mass of this hexabody bound to its antigen. And then you see that the hexabody that has like 12 arms that can recognize the antigen really binds also 12 antigens as well. Now, I don't go into the biology behind this, but I just want to show you that this Orbitrap with native mass spectrometry, with these extended mass range possibilities, can even measure masses of about 1.26 megadalton and really get accurate masses to really also measure binding to the antigens as well, for instance. Now, to make another step, and this has always been one of my favorite topics, we can also use this native mass spectrometry to look at viruses. So this is data from a couple of years ago where we analyzed different viruses, and you start to recognize the charge state distributions. From these charge state distributions, we can determine the charge, and if we know the charge, we can determine the mass. And you see here that we have like 4 different particles where we can determine the mass from, and this really ranges from about 2 million Dalton to 4.5 million Dalton. So mass spectrometry can now measure and analyze these very large particles. Now, the third one in here is AAV1, which stands for adeno-associated virus serotype 1. And compared to the other ones, it's a bit the ugly duck. It looks not so nice as the other ones. It looks way more complex. And first, of course, we were a bit frustrated. But then we started to think about this complexity of the spectrum. Can we rationalize that? Now, on the right, you see the EMR where we do our measurements on. This is maybe not how you recognize the commercial instrument. In our lab, we take off the caps most of the time because we still want to improve and modify the instrument a little bit further. But going back to this AAV, what's going on there? Why doesn't it look as good as the other viruses? That's what I want to talk about now, and I want to talk about this sizing and counting particles by native mass spectrometry. So the AAV is not just a virus. It's a very important virus because it's for us not toxic. We can actually take it and it has no negative side effects. And therefore, it has become, and it is now, an ideal gene delivery vehicle. And here you see a little bit about the biology. What people do then is they make a transgene that they incorporate into the AAV virus. And when the virus is then injected into a person, the virus gets into the cells, the virus breaks down, and then the transgene that is brought into the body can be transcribed by the DNA and can lead to protein production. This is not just science fiction, this is really happening at the moment. So it's used for gene therapy, and one of the most famous examples at the moment is Zolgensma, who used really AAV particles to deliver a transgene. Now, these AAV viruses, they are actually quite intriguing. They are quite small, and they are sort of, the viral particle, the capsid, is like a ball, and you see it over here. And this capsid contains actually 60 subunits. And these 60 subunits make up this container, this capsid that you see over here. Now, in the case of AAV, it's a peculiar story because there are 60 subunits, but it's not all the same subunits. So there are 3 different genes that make up the capsid genes, the capsid proteins, and they are called VP1, VP2, and VP3. They have different lengths that you see here at the bottom with a mass of 81, 66, and 59 kilodalton, and they assemble into the virus in total up to 60 subunits. Now, if you run a gel of these AAV particles, you see that on the right bottom here, that VP3 is more dominant than VP2 and VP1. And so people always thought that maybe the capsid distribution is 50 copies of VP3, 5 copies of VP2, and 5 copies of VP1, so that we have in total 60 copies. But is this really the case? Now, people knew already that a capsid particle, to really be functional, it requires at least some copies of VP1 and VP2. So that's important to remember. So what we now did is that we zoomed into this ugly spectrum. And this ugly spectrum you see now on the right, it's not as congested as it looked before, but we color annotated now peaks that we assigned actually to different virus particles. And we simulated these spectra, and that you see here at the bottom. And the simulations are really for a single stoichiometry. So there are 60 subunits in total, but the simulations are either for 2 VP1, 2 VP2, 56 VP3, and the second one, 2, 3, 55, etc., etc. And if the stoichiometry is different, the mass will be slightly different. And that's why you see all these spectra being shifted slightly in mass or m over z. Now then, if you have these different particles present, then of course the experimental spectrum will be the sum of these individual spectra, but a weight sum, depending on how many particles there are of that particular composition. Now the problem a little bit is in mass spectrometry, if you have such a heterogeneous sample with different charge states of different particles interfering with each other, it's a bit like interfering of different waves of different frequencies. And that's what I show on the left, just to compare it with. We can get that either spectra, when they are summed up, are positively summed up, not interfering with each other, but they can also destructively interfere with each other, and then we would maybe get gaps in our spectra. Now, in this spectrum, it looked still pretty good. We could annotate some of the stoichiometries, but there was already clearly evidence that indeed there is not a fixed stoichiometry, but it is a mixture of different stoichiometries. So the next question was, is this assembly mechanism, is that random, so stochastic, or are there preferred stoichiometries that are available for the AAV virus? Now, what we started to do is we first said, let's assume that the assembly is just stochastic. And then you can say, okay, if I have so much VP1, VP2, and VP3 protein in my cell, I start to assemble these capsid particles. Then there are all kinds of probabilities. There's a probability that I only get 60 times VP3. There's a probability that I get 5 VP1s, 5 VP2s, and 55 VP3s, and etc., etc. Now, if you know the initial concentrations of VP1, VP2, VP3, it's like taking different colors balls out of a big jar. You can calculate actually what the probability is for every stoichiometry. Now, we take this into account, and then by knowing the initial concentrations of VP1, VP2, and VP3 in the cell, we can calculate what the distribution should be, and we can simulate a native mass spectrum of that given stoichiometry. Now, you see that over here, and it's quite complicated spectra, but what I want to show is that it also depends on how long and with which high resolution, low resolution or high resolution, you listen to the signals. Now, maybe I want to focus first on the right, where if we do a simulation of the spectrum with an infinite resolution, so extremely sharp peaks, then we would get the spectrum that you see at the bottom. And every different particle has a spike. If they are very close, you can see it over here, they start to overlap. The resolution we can achieve now with our mass spectrometer is not that good yet. So if we have the resolution of 128 milliseconds and only look at this part of the spectrum, then we would record the spectrum that you see here with the green dots. However, if we listen shorter to the transient, the resolution goes down, and we get the spectrum that you see here at the top. Now, we can compare these simulations with our experimental spectrum. And that you see here on the left. And I really want you to look at the blue and black spectrum and at the green and the black spectrum. The black spectrum is each time the experimental spectrum. And especially at the bottom, we clearly see this huge interference pattern. And I must say, when we first measured this spectrum, we first thought something was wrong with the particles. But only after the simulation, we thought, no, this is as best as it should look. While beforehand, we thought, okay, maybe the 32 millisecond transient looks actually better. No, it's 128 millisecond that looks way better, but only after we know where to look for. Now, we think this is quite a challenge and quite we are quite proud that our mass analysis could analyze this. It's some of the highest resolution mass spectra of virus particles that we have ever measured. But it's also biologically relevant. We've learned now that this assembly mechanism of this important gene delivery vector is really random stochastic. So what is then the nature of each particle? Now, this is given here on this slide, and maybe I just want to show you here totally on the right. It's a distribution for each of the VP1s, so VP1, VP2, VP3, summed over all the particles that are present. So we see in the maxima of green, red, and gray, we see the most likely stoichiometric amount, but we also see that there is a distribution of it. And by looking at these pictures, we can actually say how many of the particles contain not a VP1 or not a VP2. And that's actually quite a sizable amount. But we can also say, and that in this particular preparation of AAV, we have this particular distribution of particles. Is that then always the same? Now, AAVs come in different serotypes. They are also produced in different cells, host cells. Sometimes it are human cells, sometimes it are insect cells. And growth conditions may also be slightly different. So here I have a very complicated slide, but it's actually showing AAV particles from 6 different batches, sometimes also of 6 different serotypes. And without going into much detail, the take-home message is that all these particles have a different distribution of VP1, VP2, VP3, and sometimes they are really hugely different, like the bottom 2, where in the left there's very low amounts of VP1 and VP2, and on the right there's relatively high amounts of VP2, VP3. Now, for the production process, these are very important parameters, and therefore we think that this unique way to measure these stoichiometries is of importance in the quality control of this sort of particles. Now, I want to move on, and this is another pet molecule that I really like. It's the Flockhouse virus. It's one of the biggest molecules or particles we have ever measured on our mass spectrometer. It's about 9.3 million Dalton. But you see that the quality of the spectra that we can get now for such particles is just amazing. It's really baseline resolved charge states. This particle has about 170 charges. You might say this is a lot, but it comes at M/Z 55,000. So it actually means only 1 charge per 55,000 Daltons. So I would say it's actually quite low charge. Now, this is really a genuine virus. It contains 2 different proteins of both 180 copies. It contains 2 pieces of RNA. It contains calcium. So it's amazing that we can get such beautiful mass spectra out of this. But we also realized when we measured these particles that is something quite peculiar. If we tune down the instrument, we can really now start to record single particle events. And for us, this was first a little bit amazing. And here you see that we have a mass spectrum, and in the top is an average, at the bottom is a single spectrum, and we see just these few events. Now, the first time we saw this, we thought it was already truly amazing. But if you then look through all the spectra, we also saw that these events were sort of quantized. A single ion made always the same sort of signal. So we thought that is what we expect, because the signal intensity in the orbit trap is just dependent on the charge. So if we detect a single ion, we can actually predict how high the signal should be. Or the other way around, if we can measure the signal intensity, we can directly tell what the charge is of that particle. Now, we first wanted to prove that, and we measured all these different assemblies, from antibodies to ROEL, to nanocontainers, to Flockhouse viruses. And indeed, over this whole range of masses, from 150 kilodalton to 10 million Dalton, we found this beautiful linear correlation between the signal intensity and the charge. So now we can assume that if we measure a single ion of a single particle in the orbit trap mass spectrometer, we directly, from the signal intensity, can tell what its charge is, and therefore we can tell directly what its mass is. Now, we call this charge detection mass spectrometry, and I just show by this mass spectrum that by doing this, we actually can measure mass spectra in 2 dimensions. So the one dimension is the black line, is the average mass spectrum that we take all the time. And in red is the intensity in the charge distribution. And the spectrum you see here is actually of our IgG antibody. And the IgG monomer is in blue, the IgG hexamer is in purple. And on purpose, we also have here dimers, trimers, and tetramers. And we can see that they are separated in M over Z in the normal mass spectrum, but we can see that they are even more separated in the intensity domain or in the charge domain. Now, this is already beautiful to show, but just to zoom in, sometimes two ions have the same M over Z, but another charge and another mass. Of course, this is the case with, for instance, trimers and tetramers. And here you see one example, 1060, where in the M over Z dimension, these two peaks perfectly overlay, while in the charge dimension, they are perfectly separated. So this is one example to show that this charge detection mass spectrometry gives you an extra dimension to look at particles. Now, another nice thing about this charge detection mass spectrometry, sometimes you have such heterogeneous samples that you cannot resolve the charge state. Here's an old spectrum from our lab on the norovirus, the NVLP, of about 10 million Dalton, and we could not resolve it. Here, charge detection mass spectrometry comes also to its advantage. Now, on the right, I show a little bit the workflow that we have. So we let only very little ions into the Orbitrap per scan. Not only 2 or 3, as I showed you before. Typically, we let a few hundred of these ions in, and we get a mass spectrum that you see at the top. Then we're going to look at each individual ion, and we see if this ion is a survivor or if this ion decays while it's trapped in the Orbitrap. If it decays, it actually gives not an ideal Fourier transform peak, as you see here at the bottom. And these decayed peaks, we actually reject and take out of the spectrum. So we filter those out, and we are left over with the beautiful ions that survive the whole transient time. We do this because it actually improves the resolution, and we are still left over with plenty of ions to get an accurate mass from. You see this here at the bottom. Before we have 150 or 134,000 ions. On the right, we still have 31,000 ions, but it's a much more beautiful spectrum from which we can determine the charge, from which we can determine the mass of the different particles. Now, just one example, this is a mass spectrum that if we do the conventional native mass spectrum, it didn't give us a clear answer. It turned out we wanted to measure IgM, and this is what people say often a pentamer, sometimes a hexamer. But it actually turned out that the preparation that we had was a mixture of tetramers, pentamers, and hexamers. In the conventional mass spectrum, we could not separate them. But if we also separated them by charge, we could get these 3 different species here in red, blue, and yellowish. And we could clearly separate them in the charge domain and by charge detection mass spectrometry. And with that, we could really quantify how much tetramer, pentamer, and hexamer there is. And so this is an ideal application of this charge detection mass spectrometry. Another beautiful example, we think, is where we applied it to the AAV particles. I told about these AAV particles, but I showed you only data on the empty particles. Now, of course, it's much more interesting to also be able to measure the AAV with the transgene loaded particles. Now, if it's loaded with a transgene, we don't get these beautiful high-resolution mass spectra anymore. We cannot assign its charge, so it's not that well measurable by conventional native mass spectrometry. But if we actually do it by charge detection mass spectrometry, we see in our spectrum, here in the middle at the bottom, that we clearly separate the particles without the genome and the particles with a genome. And to me, this is as close as you can get to the Coulter principle. We measure ion by ion, we size it, we measure its charge, we measure its mass, and we are now able to count how many of the particles contain a genome and how many don't contain a genome. Now, in this whole gene therapy field, this is actually a major bottleneck, a major problem. A lot of the particles, ideally you would like them all to have the genome packed, but a lot of the particles don't have the genome packed. And even more so, some of the particles have not the proper genome packed. This is very difficult to analyze by any analytical technique, so we set out to see what we could do with charge detection mass spectrometry. Again, just a little bit of background. This is what I got from a review, so it's not from our work. If people start to make these AAV particles with external genes, they sort of say, okay, we have to make a lot of enormous amount of particles. But only a small fraction of them, maybe 10%, contains the genome. And even a smaller part of that is infectious. And only a smaller part will be really transducing. So this is a major bottleneck in this field. Another bottleneck is, and if we want to optimize this better, we also have to have means to find out how many are full capsid, how many maybe have a fragment of the genome, and how many are empty. Now, techniques that are used at the moment are sucrose gradients, sometimes EM, but here we explore this charge detection mass spectrometry. So we got particles that supposedly were filled with genomes. And in our CDMS, which you see over here, we really found 3 different distributions. The distributions we could mass measure, we could count them, and here you see the counts, but we could also get the very accurate masses of these particles. Now, this was one preparation, and in orange are the empty ones, in red are the filled ones. There are quite a few filled, but we also see a distribution in the middle, the green particles, and this was already quite a bit amazing. Now, I also like to state this is single molecule mass spectrometry. So sometimes people ask me, how sensitive is this? Now, it's extremely sensitive because you need only molecules, a few molecules to measure it. So compared to what we are used to, our traditional native mass spectrometry, we often dilute our sample 100 to 1,000 times to get this data. Now, we want to explore if we could really quantify this amount of particles. And we had preparations where, on purpose, we thought we had either empty particles or fully loaded particles, or particles where supposedly 25% was full, 50% was full, or 75% was full. These ratios, they were determined by ultracentrifugation. And with these ratios, it was not taken into account that there might also be what we call now intermediately filled particles, the green particles in the middle. Now, first of all, we show here that CDMS could actually measure all these 3 different sort of particles and also quantify these particles. And our estimates of 25% full, 50% full, or 75% full was actually in reasonable agreement with the ultracentrifugation if we count the half-full particles indeed as full particles. And that's a distinction that a centrifugation could not make. Now, the extra value of the CDMS that we can also get the average mass of the particles, and we can really see that the full particles of 5.1 megadalton are indeed 1.4 megadalton higher than the empty particles, and that is in perfect agreement with the size of the genome. But we also see that the half particles probably contain something like a half genome. And this is something that we look a little bit further into. Now, the nice thing about this CDMS, and I'm really excited about it, is that we can listen to single ions. And I always ask to my students, how does the isotope pattern of a single ion look like? A single particle doesn't have an isotope pattern. So the resolution is not limited by the isotope distribution. You just measure a single particle. These particles, they are extremely stable in the Orbitrap. And so we can listen to them for longer transients. And I showed you before 28 milliseconds, 128 milliseconds. Now we try to boost that further and we listen to the transients of these particles for up to about 4 seconds at the moment. And you clearly see that we get then, even at an M/Z of 40,000, a resolution that comes close to 100,000. And I think this is in mass spectrometry really amazing that we can achieve this at the moment. Now, we use it for viruses, but it's actually the method of choice for very heterogeneous particles. Now, a heterogeneous particle that we all know about is the famous or infamous spike protein of the Susquehaff virus. Why is this so heterogeneous? And you see a picture here on the right. Because this protein, which is actually a trimer, is heavily decorated with glycosylation. And glycosylation makes this mass heterogeneity that I showed you before, makes it rather difficult for us to measure the size of these particles by traditional native mass spectrometry. But with CDMS, also with mass spectrometry, we can now get very accurate masses of these spike proteins. And you see we measure a mass of this spike protein of 477 kilodalton. The sequence of the mass, or the mass of the full sequence, is 385 kilodalton. So we really see that this spike protein has about 100 kilodalton of glycans attached to it. So also for this CDMS is very nice. That's the spike protein, and we know that we now try to target this spike protein with all kinds of antibodies. So I also want to show you that we looked at the antibody binding to the spike protein and used again CDMS to look at this binding, also mass spectrometry to look at this binding. And we found a very interesting observation. We could measure the binding of antibodies to the spike trimer. And you could expect how many antibodies do bind to the spike trimer. Is that 0? Is that 1? Is that 2? Or is that 3? And 3 is a logical choice because it is a trimer. So it has 3 RBD domains, and receptor binding domains. So maybe if an antibody targets that, you might also expect that 3 antibodies bind at the same time. But taking a whole mixture of human-derived antibodies, we actually saw that some of them bound preferentially just with 1 to a trimer, some of them bound preferentially with 2 to the dimer, and some of them didn't bind at all to the trimer. So we were using this CDMS to really look at the different binding affinity of these molecules and different modes of binding of these antibodies to the spike trimer. So the particles that we measure, you see roughly on the right, and you might now say, why are there not 3 antibodies bound to the trimer? Maybe this picture tells more than what I can say. You see that the size of the antibody, here in blue and orange, is about the size of the full trimer spike protein as well. And you can easily think about how maybe 3 antibodies would sterically clash with each other if they want to bind to such a spike trimer as well. Now, we also showed in collaboration with our collaborators that you don't need 3 antibodies to bind to also be potent in neutralizing virus infection. Even the binding of a single antibody to just 1 trimer is for some of these antibodies already good enough to get real neutralization. So we can use this CDMS for many different things. And I think it's an important new toolbox for mass spectrometry because we are facing this ever-growing field of more complex biopharmaceuticals. And these next-generation biopharmaceuticals pose new analytical challenges where we think that high-resolution native mass spectrometry can provide the required details. The single particle charge detection mass spectrometry on an orbiter provides unique sensitivity and allows us to analyze very complex, structurally complex, large biomolecules, as I hope I have showed you here, with viruses or vaccines, but also these highly decorated glycoproteins. So with that, I come at sort of the end of my part 1. And before I move over to the second part, I just make a very small break. Okay, to some of you, welcome or welcome back. Now we'll shift gears and talk about a second part of our current research. And again, being a mass spectrometrist, I would never have thought that I would ask this question to my audience. How unique is your immune response? Because I'm not an immunologist, I'm an analytical chemist, I'm a mass spectrometrist. But we got interested in this, and I just want to show you what sort of data we have been recording lately and what sort of new biology and maybe some new immunology we find by this. So we are interested in looking at antibodies. We are interested in looking at antibody repertoires. Now, antibodies are a beautiful sort of molecules. They are big, 150 kilodalton. And of course, in our blood, a quarter of the proteins are antibodies. So it's really an important part of our proteome, of our plasma proteome. But of course, it's also an important part of our immune system because the antibodies are there to fight pathogens, viruses, bacteria, but also other human diseases. And they all come from our own system, from our own immune system. So I am quite a bit intrigued by these antibodies, and I wanted to look at these antibodies already for quite a long time. So if you look at our blood, and if you look at our plasma, then it contains quite a bit of protein. Now here's a pie chart of which proteins are in our plasma based on their concentrations. So about half of our plasma proteome consists of the protein albumin. But about a quarter of our plasma proteome contains this IgG. And IgG is just one of the subclasses of antibodies, as there is also IgA, which you maybe also see in this pie chart, and IgM. Together they are our antibody repertoire. And as you see here, they are the second most dominant protein in our blood. So, is this just one molecule? Now, for albumin, that's sort of true. But for IgG, every person can make all kinds of different IgG molecules. To explain that, I also show here the structure of an IgG molecule and how it is built up from different segments that are created by different genes on our DNA. And we are actually making these antibodies from different genes. And then these gene products, they are shuffled in such a complex way that we can, from a few building blocks, make an enormous repertoire of antibodies that are in sequence slightly different from each other. And because they are slightly different from each other, especially in what is called these variable regions, CDR1, CDR2, and CDR3, and that are the regions that are really recognizing antigens. That are the regions that really target the pathogen, neutralize the pathogen, like the coronavirus, for instance. And they do that by having a unique sequence that recognizes the epitope on the antigen that they are interested in. So how many antibodies and how many antibody sequences can a human person make? This number is quite incredible. Every antibody is made by a B cell. And you can calculate that there are an enormous amount of B cells in our body. And there are already 10 to the 11th total number of B cells in our body, and every B cell has a different mutation of the IgG, and therefore can produce a slightly different IgG molecule. Now, this number of 10 to the 11th is maybe the number of different antibodies we can make, and that's an enormous amount of number. If you put that into context, people normally say we have only 20,000 genes. But we have already cells in our body that can make 10 to the 11th slightly different IgG genes that all lead to a slightly different antibody, but functionally maybe with a very different pathogen that it can target. So our body and our repertoire of different antibodies is and was thought to be staggering. And maybe we can make 10 to the 9th, 10 to the 11th, or maybe even 10 to the 18th different antibodies in our human system. That number is really amazing. But that we can make them by our B cells, does that also mean that we really are making them? And does that mean that they are really circulating in our blood? So this was the question that we set out to do. How many different antibodies are really present in our blood, making up this quarter of all the proteins in our blood? Now, what is this hypervariability? And I just want to show this here. It is the sequence of the change in the antibody that are really these hypervariable parts. And we see that certain amino acids, they are quite constant, and that's when you see a full letter, while certain parts, they allow different kind of mutations, and here you see sort of the frequency of the mutations at the sites. But you can imagine that if you take this all together, the combinatorial sum of all these modifications, that leads to these millions to billions to zillions of different antibody sequences. Now, as a mass spectrometrist, when you look at this, then you say every mutation that you see over here leads to a slightly different mass of the antibody. So maybe every antibody has a slightly different mass. And that's sort of what we take into account when we use our mass spectrometry-based approach. So what did we do? It's relatively simple. We start with blood. We capture all the IgG molecules out of the blood. And then we do something a bit clever. We cleave off from the antibody only the region where these hypervariable parts are, only the region that is recognizing these antigens, and we get rid of the major part of the molecule, which is called the Fc part. And where we are left over with is called the Fab fragments. And these Fab fragments, they harbor these CDR3 regions, and they harbor all the information that make a clone, and that's how we call them, make an IgG clone derived from a single B cell, unique and different from another IgG derived from another B cell. So then we brought all these Fabs, we collect all these IgGs from serum, we produce all these Fab molecules, and these Fab molecules have a mass of about 45 kilodalton. We cleaved off a major part of the rest of the molecule. We bring all these molecules to the mass spectrometer and we just subject them to LCMS. And that's what you see over here. This is the LCMS trace that we get. Now, if this contained a zillion different antibodies, we would just see a big blob. But the LCMS trace that we measured is really showing us specific features. And if the animation works, yes, here it goes, we walk over this LCMS trace and we measure each time the mass of the ions that come out. And these are all Fab molecules, and they all have masses of about 47,000, 45,000. But each time the mass is about a few Dalton difference because the sequence is slightly different. So with this, we go over this whole LCMS trace, and our mass spectrometer allows us now to identify the mass and retention time of each clone. And we say then, okay, the antibodies that we see, we can type them by their retention time, and we can type them by their molecular weight. And if they have a unique mass and a unique retention time, we call this a clone. So how many clones are then present in such an antibody repertoire? We can now quantify. Now we can also show these plots differently, and this is sort of a mass plot. And we leave out the retention time information here. But this is a mass plot of the clones that are present in the plasma of a certain person. And we see it's dominated, maybe by 50 peaks, maybe by a few hundred peaks, because every signal you see here is not noise, it's really a different antibody with a different mass. We can also quantify these antibodies because we spike in a monoclonal recombinant antibody. So on the y-axis, you really see what the concentration is of these individual clones in the plasma. Now, for us, this was a big eye-opener. We can measure a repertoire of IgGs, and in this case, it's IgG1. We can measure the repertoire of IgG1 in plasma of a single donor. Then we set out the following experiment. We said, if we have a single donor, can we get blood now? Can we get blood in 1 month? And can we get blood in 2 months? And we also set out to do the experiment, can we get the blood of another person and measure this profile at 0 months, 1 month, and 2 months? Now, the results are on this slide, and you can actually see that the repertoire of the red person is very constant over time. It's maybe also because it's a healthy person. So someone has a certain antibody repertoire in their blood, and this repertoire remains relatively constant over time. The blue person also has a repertoire that remains relatively constant over time. The next question that we asked, but how many of the clones of the red person do we also see in the blue person? Not only in numbers, but also in total concentrations. Now we depict that here in this heat map, and you clearly show that there is close to zero overlap in the clonal repertoire of one person over another person. And we can still get the unfortunate occasion that they have a clone with the same molecular weight and retention time, because our resolution is maybe not perfect yet. But we actually think there is a clonal overlap of 0 between two individuals. So the main big message over here is each person has a unique repertoire of antibodies, a unique repertoire of IgG molecules. Now, the next experiment that we did is that we said, okay, if we can follow repertoires over time, can we maybe look at patients that have had a real physiological change? Now, here we had samples of 8 patients that had undergone surgery, and unfortunately for them, they got an infection, they were brought to the ICU, and fortunately for us, unfortunately for them, they all survived. Fortunately for us, of these people, at 4 time points, blood was taken. For the operation, when they developed sepsis, also a bit later after while they were still at the ICU, but also at the discharge of the patient. I won't go into much detail, but we measured these clonal profiles, and the clonal profiles of a given person remained unique, remained relatively stable, although there was also dynamics in the clones. Some clones came up in intensity, some clones went down in intensity. And of course, the clones coming up may be antibodies that are only produced when the person has seen the pathogen that caused the infection. So we are interested in these antibodies because if these antibodies are targeting the pathogen of infection, it may be, in principle, be a lead for a new biopharmaceutical, for a new antibody therapy. So we can now monitor the dynamics of each individual clone in each individual person. Now, first of all, we found also in these 8 people that there was almost no clonal overlap between patients, but we found now a bit more dynamics within each person, because especially at time point 2 and 3, the clonal repertoires in some individuals changed quite dramatically over the first time point, while in other persons it remained, like in this male 67, they remained a bit more constant, as shown here in this heat map, but also in this hierarchical clustering. Now, we had a particular donor whose antibody profile was very simple. Not only that, you see that at the top trace, only a few peaks are present, but we also saw that this person, after being infected, developed one antibody to an enormous amount at T2, T3, and T4. So we hoped and we thought this is an interesting clone, because this clone may tell us a little bit about what happened in the infection, and maybe this is an antibody that can target the pathogen that has caused the infection in this patient. So we targeted this antibody and we said maybe we can derive its sequence, because now we only know its molecular mass, we know its retention time, but we don't know anything yet about the intensity of this antibody. So to do that, we did top-down mass spectrometry, getting the full sequence of the FAB. But we played also a little trick that you see at the bottom. We also reduced the sample, and then the FAB molecule is broken in 2 chains, the light chain and the FD part. And you see that this reduction works quite well. We get 2 new peaks, and we get also 2 new peaks for the green FAB. But that one we were not so interested in, because that was already present before the sepsis episode. Now, what we did, and this is quite complex, but we wanted to sequence this antibody in this background of hundreds of other antibodies that almost have the same sequence. I can only tell you, and I hope you believe me, this is extremely complex, because the background is as alike as it can be, with only changes in the CDR region. So measuring, sequencing an antibody in a background of plasma is almost impossible. Now, here we were lucky, because the antibody of interest was by far one of the most dominant antibodies in the plasma, and that helped us to do sequencing of this antibody by top-down mass spectrometry, also by bottom-up mass spectrometry, using different proteases, And by combining all this data together on the Fab, on the light chain, on the FD, on the peptides that we made with these different proteases, we derived, and this was quite a task, to the full de novo sequence of this antibody. Now, if you have then this sequence, you still have to prove that this sequence that you derived from the mass spectrometry data is correct. But then here you see some more data of how we annotated this sequence. And you always see that with top-down on different parts, you never get full sequence, you always get sequence of different regions. So you really need all the data together to get the full sequence. Now then we had the full sequence and we made the recombinant antibody. The recombinant antibody had the same retention time. You see that here on the first window as the clone that we were interested in. It had the same mass and the same mass spectrum as the plasma clone. And most importantly, it showed exactly the same fragmentation and therefore exactly the same sequence as the plasma clone. Now, we could also confirm that by looking at the peptide data where you see the mirror plots. And so this was our validation that we know now that we derived the clone from plasma, we derived its sequence, we made the synthetic recombinant antibody, and we know for sure that the sequence we had was correctly done. Now, for us, this was a proof of concept. We can do this now in many more applications. And just to end, I want to show you what sort of applications we are doing now, just by maybe telling you that this work was just published in December in Cell Systems. And I think it's a very nice approach to look at IgG repertoires. Now, one way to look at it is a paper that we also contributed to already, where we looked at the IgG repertoire actually in human milk, but also in human milk of mothers that have had a COVID infection. And now we measured the profile of the milk antibodies, but we also measured the profile of the antibodies that had been treated with the spike protein by depleting then only the IgGs that bind to the spike protein. Now, we measured the profile after depletion that you see here in green, and we measured that the profile was exactly like in blue, which is the original profile, but some peaks were depleted. Then we started to elute what was bound to the beads, and in this way we found that this particular clone in the milk of the mother is really a clone that binds to the Susquehaff trimer, the spike trimer that I mentioned to you before. And now, of course, we want to sequence this antibody as well, and maybe see if it can be useful in therapy as well. Now, I showed you already that we can do this in blood, but we can also do this in human milk. And this is another recent study that we did in this work, monitoring, which I found very exciting, the antibody repertoire, the IgA1 repertoire in two mothers during the lactation. So we had over a period of 16 weeks, we had these mothers provide us samples almost every week. We measured their profiles, and the message was exactly the same. Each mother has its own unique IgA profile, and this profile doesn't change that much over time, unless, and this was the case for one of the mothers got a little infection, and then we saw certain clones coming up in this mother as well. Now, I hope with that I have shown you that mass spectrometry is a very powerful technique, that we can learn all kind of new biology about that, and especially this field where we go into measuring antibody repertoires that you make yourself, that we try to find out which are the best antibodies that a person makes against a certain pathogen. This is something that we in the lab want to work on for the next years, to really try to help society by finding new antibodies that are made, as I said again, by humans, can maybe be used in other humans to fight pathogens that we have to fight with all of the time. So I find our body very beautiful. I also look at it as a source of antibodies. I hope I've told you now that each person makes its own repertoire of antibodies. And in this enormous resource, not only of a single person, but of all people in the world, I'm pretty sure that there are ideal candidates for biotherapeutics. And what I'm also pretty sure about, that the approach that I presented to you here today may help us and may guide us to find the best candidates for new medicines in the future. With that, I'd like to end this part. I thank here the whole lab. It's a big lab in Utrecht with a lot of people from all over the world. I also like to thank all the collaborators and all the funding agency. And I'd like to thank you for your attention. And with that, I give the second break. And I hope you stay on, or you take later a look at part 3, where we, especially about this last piece of work that I described in this lecture, and made together with some artists, which I think is a beautiful video. So I hope you will enjoy that as well. And again, I thank Pittcon, I thank the Wallace Coulter trustee for this honor, for presenting this lecture, giving me the opportunity to present this lecture. And with this, I'd like to end. Thank you very much. Welcome. Welcome back maybe to the third part of my lecture. And this is really something that I did with a lot of pleasure. Science is sometimes complicated. Science is sometimes difficult. But I always like to talk about science to as many people as possible. And of course, you do that because you want to make them as enthusiasts for the science that you're doing as yourself. But I also find that the feedback you get, even from people that don't know that much about the subject as you do, can be amazing and can actually lead to new things that you want to explore and can also give you new insights. Now, what we have been doing over the last half year is that we got in contact with animation artists, and we got in contact with dancers from a group in the Netherlands. And together we talked about our science and we talked about their art, and we decided to make a movie together. And this movie is all about antibodies, the human source of antibodies. And I really like the connection with the dancers, because I see antibodies as some of the most flexible molecules in our body that can interact with whatever they want to interact by dancing around, by making connections, and by targeting, as they are called, antibodies, by targeting the antigens. So the movie that I hope you will enjoy as well was made in collaboration with artists, video artists, and dancers. And it tries to show how our own body has its own unique repertoire of antibodies and how we can study that and what we can learn from that. So I hope you enjoy it. Let's start.
Antibodies. Proteins made by our body to kill foreign invaders. We are constantly under attack from bacteria and viruses. And at the same time, we also face threats from within, such as emerging cancer cells. While some of us won't even notice these attacks, others can get seriously ill. This is due to the uniqueness of our immune systems, including our body's individual ability to make the right antibody to deal with a specific threat. The powerful and sophisticated human immune system can make quintillions of slightly different antibodies. That's billions of billions, a nearly infinite resource. When one of these antibodies turns out to be successful at its job, the body starts making more and more copies. As a result of the lifelong training and adaptation of our immune system, what appears to be a simple trial and error system is in fact something far more clever. At any given moment, we only make those antibodies that are really needed, meaning that only about a couple hundred or thousand will ever be dominantly present in our blood. To tackle diseases, human antibodies need to be as unique and flexible as dancing human bodies. What if we could discover which antibody fights which threat the best, and then turn these antibodies into biotherapeutics? We could then use them to treat patients who are not making the right antibodies or not making enough of them. This would bring us close to something that represents a cure for just about every disease. a cure that stems from within our own body. Using mass spectrometry-based protein analysis, we can now directly monitor the repertoire of antibodies present in blood. Just a few droplets is enough for us to measure how a person is responding to an infection or vaccination. As a result, we can search for specific antibodies present in those who do not get ill, enabling us to identify the optimal weapon against the disease in question. The quest for the most powerful antibodies is only just beginning, but the solution to any disease might just come from within you.
I hope you enjoyed the video. It's actually a sort of premiere of the video. It was just finished a month ago. It was also partly made for this Wallace Culture lecture. I'd like to end again by thanking you for being present, for listening to my presentation, for watching the video. And I'd like again to thank the people of Pittcon, and especially the people of the Coulter Award. It has been a great pleasure for me to be the awardee. It has been a great pleasure for me to work with all of you. Thank you very much.